Thank you to organizers for giving my presentation actually the place after Marta presenting not published EULAR recommendations. You know, it's a difficult task to follow such an excellent presentation. I will try. So the title of my presentation is What Can We Learn from Monogenic SLE? In my presentation, I would like to focus on the biology behind the disease. So what can we all learn from the most severe refractory cases? And on the studying pathogenesis in these monogenic severe cases to somehow transfer this knowledge in more common disease patients. First, I would like to start with this slide showing how we could classify pediatric, I'm a pediatric rheumatologist, so pediatric inflammatory non-infectious diseases. You can see on the far spectrum of this cartoon that we have monogenic diseases of myeloid cell lineage that are causing autoinflammatory diseases, and on the other side are monogenic diseases causing defects of lymphoid cell lineage that are causing autoimmunity. You can see that SLE is a little bit closer to adaptive immune defects. Most of our polygenic diseases can be somehow found in between, but in my presentation I would like to focus on monogenic diseases, and I would like to show you that SLE is really a heterogeneous disease. Let me start with inborn errors of immunity. We know that there are more than 500 diseases that are causing inborn errors of immunity, and in nearly half of these diseases, we could see some autoimmune or autoinflammatory phenomena. And definitely these inborn errors of immunity provide a unique insight into the mechanisms of autoimmunity. Together with a colleague from our unit, Mark Kachar, we tried to summarize what are the mechanisms of autoimmunity in inborn errors of immunity. You can see here the different mechanisms, and in let's say the mechanisms that are of particular relevance for SLE that I would like to somehow present a little bit deeper is type 1 interferon hypersecretion that we face in several levels. Gain of function mutations in T and B cells activations, such as STAT3 gain of function mutation. And the second part of autoimmunity as a part of inborn errors of immunity that I would like to particularly focus is defective clearance of immune complexes and apoptotic cell bodies, not just complement deficiencies, but also monogenic defects of apoptotic mechanism. So with this background knowledge, I would like to move now on childhood onset SLE. This was a nice study, epidemiological study that our colleague from Toronto, Linda Hiraki, made when she analyzed the US Medicaid data when SLE presented. And you can see in this slide that lupus very rarely presents in children younger than 9 years of age. But in those that present before 10 years of age, it is very likely that there is some underlying monogenic disease. So what is SLE? What are the monogenic diseases that I would like to present in a little bit more details? Complement deficiencies, apoptotic defects, genetic overproduction of interferon alpha, defects in adaptive immunity, and defects in metabolic pathways. This is a cartoon that were made by colleagues from France. What are the kind of a gene distribution in monogenic childhood onset SLE? Complement deficiencies are lacking here. Complement deficiencies are the most common. They are causing 1/3 of monogenic SLE. You can see that this is followed by apoptotic defects. The most common is defect of DNase 1L3. The various defects in the red color, the type 1 interferon defects. In green color, you can see monogenic defects in adaptive immunity. The most common is protein kinase C delta deficiency. And you can see that also some pure metabolic defects can give us the clinical phenotype that we know under the name of systemic lupus erythematosus. This is a study done in Turkey. They analyzed for nearly 50 patients with childhood onset SLE with early disease onset in Turkey. And they found pathogenic variants in 9.5 of patients with early onset disease. Just recently at the press congress, French group presented the analysis in their cohort, and they reported that 11% of childhood onset SLE had some monogenic underground. This number is going up. What I would like to show here that when you are doing the genetics in childhood onset SLE, not every patient has pathogenic variant. You can also find variants of unknown significance in genes that were associated, and I would particularly like to highlight the gene IFIC1. At the end, you would see that they kind of report it as a variant of unknown significance, and I will show you one of our patients with gain of function in this IFIC1 gene. So what are the clinical patterns in monogenic SLE? Depending on the gene that is behind the disease, also the clinical phenotype of SLE differs. You can see that in apoptotic defects, so this is the first column, the apoptotic defects, renal failure is present in almost every patient. So at our unit, we have 4 patients with apoptotic defects. 2 of them had end-stage renal disease. We are considering renal transplantation in one of those patients. In type 1 interferonopathies, the neurological involvement is very common because interferons actually are devastating for development of our CNS. And those that have underlying inborn errors of metabolism have more hepatosplenomegaly and more infections, because these are mainly metabolic diseases. And to put this together, the clinical phenotypes linked with these monogenic variants, you can see that systemic lupus erythematosus is not a single disease. I would say is 100 different diseases with different backgrounds, particularly if we are learning from monogenic SLE. So now I would like to dive a little bit deeper after this kind of a general introduction. And I would like to move first into the defects of complement. Complement is extremely kind of an interesting protein. It represents 10% of globulin fraction in our serum, 10%. This is an enormous amount. It's the only protein that has more regulatory proteins than the concentration of regulatory proteins of complement are higher than the complement itself. Why? Because it has completely different mode of action than our adaptive immune system. Adaptive immune system waits to recognize something and then acts. Complement reacts to everything that is not recognized as self. So it's a big difference. So it is kind of constantly in a state of activation without of these control proteins. So the functions that I would like to somehow particularly highlight is complement function in opsonization and processing of immune complexes. Complement has a dual role in the development of clinical picture of SLE. We know that complement is devastating for SLE, right? It causes inflammation, it causes tissue damage. So if you would have monogenic defect of complement, how come that this is associated with SLE? But this is the case actually that also too little of complement can cause the clinical picture of SLE. You would have failure of clearance of damaged tissue. So apoptotic antibodies would not remove, immune complexes would not be removed, and this would cause autoimmune response. So we do know that loss of function mutations is C1QRS, C4, C2, so this early classic pathway complement activation would cause clinical picture of monogenic SLE. And it is a impairment, monogenic impairment of innate immune mechanism. Complement is part of our innate immunity. So in this view, you can regard this variant of SLE as a monogenic autoinflammatory disease. It's a monogenic defect of complement. Complement does not have function only in impaired clearance of immune complexes and apoptotic cell debris. It is also somehow involved more proactively in the functions of C1q on interferon alpha production, and it also can alter negative selection of self-reactive B lymphocytes, so is directly acting on adaptive immunity. So you can see that it has several spectrum of functions. So this is a nice cartoon prepared by the colleague from Liverpool, Christian Hedrich, that shows that on one side you can see at the top complement deficiencies as a monogenic autoinflammatory disease as a cause of monogenic SLE, but now I would like to move on the completely other spectrum, on a monogenic defect of adaptive immunity, causing also the clinical phenotype of systemic lupus erythematosus, and I would move to apoptotic defects. The most common is DNase L3 deficiency. What is DNase 1L3 deficiency? It is extracellular DNA degrading enzyme. So all these extracellular microparticles that contain DNA chromatin from apoptotic antibodies, apoptotic bodies, needs to be degraded. And this DNase 1L3 is cutting nucleic acid, just cutting. And you can imagine that this DNase 1L3 is present at the highest concentrations in cells that are involved in this apoptotic process. So mononuclear cells, macrophages, liver cells. And if you have defect in apoptotic mechanisms, you would have an accumulation of a free circulating DNA that would cause the production of antibodies against this DNA, so you will have positive anti-DNA antibodies. And this free DNA would be sensed by our endogenic nucleic acid sensors that are receptors involved in interferon signaling. So you would have promotion of interferon signaling, interferon production, if you have defect in apoptosis. So clinically, this is associated with hypocomplementemic urticarial vasculitis, lupus nephritis. I would like to present one of our cases. Boy presented at 1.5 years of age. You can see that it's really with apoptotic defects, tendency for early onset with systemic inflammation. He had recurrent episodes of urticaria at the beginning, arthritis, quite severe abdominal involvement with poor weight gain, abdominal pain. His lab showed low C1q, positive anti-C1q antibodies, and he had a skin biopsy that showed leukocytoclastic vasculitis and was diagnosed with hypocomplementemic urticarial vasculitic syndrome. Later, he developed the systemic inflammation with fever episodes. He was treated with azathioprine, MMF, cyclophosphamide, I would say with partial defect. Then he developed this hemorrhagic vasculitic rash that changed over time. We did genetic analysis, found that he has a DNase 1L3 gene mutation. You can see here circled. And together with colleagues from France, from Lyon, Alex Belot group, we combined our cases with DNase 1L3. So 3 of our patients are included in this cohort, and you can see on this slide that these patients can develop urticarial rash. They can have this polymorphous rash associated with systemic inflammation. They can develop full house lupus nephritis with immune complex deposition. You can see also crescent formation here. And we were particularly interested in this study to show what is the interferon signature. On the far right, you can see interferon signature in type 1 interferon monogenic defects. It's very, very high. In patients with DNase 1L3 mutation, it is not so high, but it is increased compared to healthy control. And our patient is now, I would say, partially responsive to rituximab and JAK inhibitor. He's getting tofacitinib, and if he will continue to have active disease, we would consider bone marrow transplantation, because this would remove the autologous, allogeneic, not autologous, bone marrow transplantation. So next, I would like to move to the large group of diseases associated with genetic overproduction of interferon alpha. So interferon alpha can be influenced also by our other monogenic defects, but I would like to deep dive into these monogenic interferonopathies. A few years ago, with colleague Aleš Goropevšek, a very nice, excellent flow cytometrist from our lab, we prepared this cartoon on JAK-STAT signaling pathway. I would just like to highlight, I do not have a pointer, but just, so after binding of cytokine, you have this dimerization of the receptor, docking of STAT molecules, phosphorylation, and moving to the nucleus. In addition to this JAK-STAT pathway, I would like to particularly highlight in a red color SOCS protein. It is, the name says it all, is a suppressor of cytokine signaling. In this JAK-STAT pathway, you have also a lot of suppressors, inhibitors that are inhibiting this overactivation. And these are important because you can imagine that in our body, we can have monogenic defects for almost every protein. We just kind of need to search deeper. So defects in JAK-STAT signaling pathway, we know that loss of function mutation would cause hyper-IgE syndrome, which is an immune deficiency, but gain of function mutation in STAT3 would cause a clinical phenotype of SLE with autoimmune cytopenias, several autoimmune manifestations. And for type 1 interferonopathies, you can see on the left-hand side, we have sensors of DNA, hybrids DNA, RNA, and RNA. So these are sensors of endogenic nucleic acid, and you can have in different parts of this signaling cascade monogenic defects that are causing enhanced interferon production, and partially, this could be blocked by JAK inhibitors. And when I say partially, I would like to share with you one of our patients who presented at 15, so he is now 15 months of age, but he presented at 2 weeks of age. He had some neurological manifestations, cytopenia, leukopenia, thrombocytopenia. His infectious workup was negative, and his kind of a CNS evaluation showed that he had actually calcifying microangiopathy. With this neurological involvement and MRI sequencing, we suspected that this could be kind of a interferonopathy, this early onset interferonopathy. This is initial MRI. You can see already some changes in the basal ganglia area. We performed interferonopathy workup very early. So this was within first months of his life. We found extremely high interferon alpha concentration in serum, extremely high, even higher, let's say 3 times higher than in serum was in interferon alpha in cerebrospinal fluid. And whole exome sequencing was showing that he has a gain of function mutation in IFIH1 gene, the gene that I was showing you before in the Turkish cohort. It has been described linked with monogenic SLE. And IFIH1 gene is actually the gene that encodes MDA5 protein. And you know that anti-MDA5 antibodies are antibodies that are linked with juvenile dermatomyositis with interstitial lung involvement. So it's interesting how these diseases are perplexed. And some of the interferonopathies present with interstitial lung involvement, like SAVI syndrome. So it's not a coincidence, definitely not. This patient had a genetic confirmation, I think was 6 to 8 weeks of his life. So very early he had a genetic diagnosis. We were quite happy to find this paper in New England Journal of Medicine showing that baricitinib, JAK inhibitor, in this type of Aicardi-Goutier would be a targeted therapy. We discussed with family. We said, okay, in this study published in New England Journal of Medicine, patients were started at the median age of 3 years. So we said, okay, this boy is 2 months old. We would expect really remarkably better outcome. We checked JAK-STAT signaling in this patient. On the left-hand side, you can see the before and after baricitinib. The total STAT1 did not change, but phosphorylated STAT1 actually after 15 minutes of interferon alpha stimulation, after baricitinib, significantly reduced. So we were quite happy that actually it showed that this could be somehow beneficial for this patient, but neurologically he was not improving. We were almost crying when we saw this MRI after 1 year of follow-up, which showed that he is developing anencephaly almost. So interferon is like a bump in the brain, and JAK inhibitors are working nicely in the periphery. So his cytopenia improved, his skin rash improved, but neurologically he was kind of progressing. So it's a lot to learn also in this group of diseases. And I would just like to show you this. I already mentioned SOX mutation, so suppressor of cytokine signaling. You can see that these patients have high expression of STAT1, STAT5, and clinically they also presented with the phenotype of SLE with autoimmune cytopenia, glomerulonephritis. What was somehow encouraging in this study that with ruxolitinib therapy in SOX1 haploinsufficiency, you could see actually suppression of YAG-STAT signaling and also some improvement. Just briefly, I would also like to share somehow our data on STAT signaling in childhood onset SLE. We did show and publish this a few years ago in Journal of Rheumatology, higher expression of STAT1, STAT5, higher also basal phosphorylation of STAT1, while basal phosphorylation of STAT5 was comparable, but there was less response to stimulation. We deep dived and looked deeper, not just the STAT signaling in the mononuclear cell population, but we looked specifically in different subsets, and we found that particularly in CD4 cells, there is a subgroup of patients actually with a ratio of actually the ratio of lymphocyte T conventional and activated Treg cells. Those patients that had this ratio high had a worse disease kind of a progression. They had significantly more flares, which is showing that it's one of the intrinsic mechanisms in SLE patients. Particularly, actually, the reason is the low activated Tregs in a subgroup of patients. And what we showed that this actually, the ratio was associated with disease severity regardless of the treatment that patients received. So actually, it was a surrogate marker of disease severity. Just to show, yes, we know there were baricitinib trials, phase 2 clinical trials, and phase 3 clinical trials. I do not have time to go deeper, but I would like to just present also the last mechanism that I would like to share with you as a cause of monogenic SLE, which is a defect in adaptive immunity. It's a protein kinase C delta deficiency. This is kinase that is involved in B-cell apoptosis and proliferation. And if you have defect in this kinase, the B cells will proliferate. The thing is that not functioning B cells will proliferate, but autoreactive B cells will proliferate. And of course, this would lead to autoantibody production. It was interesting that at the same time, rheumatologists published in Arthritis and Rheumatology a monogenic defect in protein kinase C delta deficiency as a cause of monogenic SLE, and you can see the typical clinical features of lupus. And immunologists at the same time published in Blood the same genetic defect as a cause of new monogenic variant of common variable immune deficiency, CVID, which can also cause autoimmunity. But it is showing that sometimes we just need to communicate. And on the left-hand side, you can see flow cytometry results in healthy donor and in a patient. You can see, if you look at the CD27, there is almost absence of CD27 population in a patient. These are memory B cells, which are completely absent almost in patient. And a very important subset of B cells is the CD21 low B cells, which are autoreactive B cells. And in a patient, you can see in this square the population that is completely absent in the healthy donor. So the CD21 low population, and also on the right-hand side, you can see how increasing is this population in the patient. So based on this, I wanted to share with you some kind of a knowledge that on one side we have autoimmunity, on the other side autoinflammation. It's perplexed. And also on one side we have immune deficiency, but on the other side immune proficiency, and you could find examples of monogenic SLE in every part of this spectrum where you moved. So at the end, I would like to conclude that monogenic variants of inborn errors of immunity are really giving the whole spectrum of clinical phenotype of SLE and are teaching us the pathogenesis of autoimmunity and inflammation. We do believe that every child with SLE needs to have genetic testing. I strongly believe that in the future we will be able to better classify these patients. Why is this important? Because with our new treatments, we do believe that we will be able even to provide more targeted treatment if we would know the pathogenic mechanisms in our patients. So at the end, I would just like to thank our funding agencies, my colleagues, and of course, I would like to invite you to the Congress in Ljubljana. Thank you very much.